In today's video, we shall discuss about hybridoma technology and its applications. But before that, do subscribe to my channel and let me know your opinions and suggestions in the comment box. Hybridoma technology is one of the most common methods used to produce monoclonal antibodies. In this process, antibody-producing B lymphocytes are isolated from mice after immunizing the mice with specific antigen and are fused with immortal myeloma cell lines to form hybrid cells called hybridoma cell lines. These hybridoma cells are cultured in a lab to produce monoclonal antibodies against a specific antigen. This can be achieved by an in vivo or an in vitro method. It is preferred above all the available methods to produce monoclonal antibodies because antibodies thus produced are of high purity and are highly sensitive and specific. Hybridoma technology was first developed by Georges Kohler and Cesar Milstein in 1975, which led to the Nobel Prize in Physiology or Medicine in 1984. Before moving on to the details of hybridoma technology, we shall first see the difference between monoclonal and polyclonal antibodies. Monoclonal antibodies are a homogeneous mixture of antibodies that are monospecific in nature. These antibodies have affinity and specificity towards one epitope of a selected antigen. Polyclonal antibodies are immunoglobulin molecules created by totally different B-cell lineages, and these polyclonal antibodies react against multiple epitopes of a particular antigen. Polyclonal antibodies are heterogeneous mixtures of various antibodies with the potential to identify multiple epitopes. Steps in hybridoma technology. Following are the major steps associated with hybridoma technology: immunization, isolation of B lymphocytes, preparation of myeloma cell lines, cell fusion, followed by selection and screening of hybridoma cells, and finally cloning and propagation of hybridoma cell. We shall now see the steps in detail. Step 1. Immunization. The first step involves injecting the laboratory animals like rabbits or mice with a selected antigen against which the antibodies are raised through a series of injections over a period of several weeks to stimulate B-cell differentiation into plasma B cells and memory B cells. Once a sufficient number of antibodies are created in the animal serum following a few weeks of immunization, the animal is sacrificed. Step 2. Isolation of B lymphocytes. Following sacrifice, the spleen is removed in aseptic conditions to isolate the activated B cells. This procedure is performed using density gradient centrifugation. The presence of antibodies in the serum is identified using methods like ELISA or flow cytometry. The serum contains the activated B lymphocytes that produce antibodies. The activated B lymphocytes are then fused with myeloma cells. Step 3. Preparation of myeloma cell lines. Few weeks before the cell fusion, metastatic tumor cells are incubated in 8-azoguanine to get non-functional hypoxanthine guanine phosphorybosal transferase (HGPRT) genes in the myeloma cells. Non-functional HGPRT can stop the assembly of nucleotides from the salvage pathway and makes the metastatic tumor cells sensitive to HAP media as the preferred method in hybridoma technology. Step 4. Cell fusion. Cell fusion is the process in which the activated B lymphocytes are fused with HAT-sensitive myeloma cells. This step is performed by centrifugation of freshly obtained activated B cells with HAT-sensitive myeloma cells in a fusion-promoting media. Polyethylene glycol is used in this procedure. PEG helps in the fusion of cells by promoting the fusion of the plasma membrane of the myeloma cells with the plasma membrane of the antibody-producing cells, thus giving rise to a cell with more than one nucleus, forming heterokaryon. Another method used for fusion is electrofusion, in which cells are fused under the effect of an electric field. This method is more efficient than the previous method. Step 5. Selection and screening of hybridoma cells. The B lymphocytes contains HPRD1 gene which codes for enzyme hypoxanthine guanine phosphorybosal transferase, HGPRD. The enzyme HGPRD involved in synthesis of nucleotides from hypoxanthine present in culture medium. Therefore, B cells can grow in medium containing hypoxanthine aminopterin thymine. But myeloma cell lack HPRD1 gene, so it does not produce HGPTR enzyme, and it does not grow in HAP medium. The myeloma cell fused with another myeloma cell or those do not fused at all die in HAP medium since they do not utilize hypoxanthine. Similarly, B cell that fuse with another B cell or those do not fuse at all die eventually because they do not have capacity to divide indefinitely. So, only hybridoma cell, which is the fused cell between myeloma and B cell, can survive and divide in HAP medium. Screening is done to select hybridoma cells, which are the desired cell for monoclonal antibodies production. The methods for screening hybridoma cells includes ELISA, which is a widely used method to assess the specificity and affinity of antibodies produced by hybridoma cells. Western blotting, which is used to assess the specificity of antibodies by detecting their binding to target proteins separated by electrophoresis. Immunofluorescence, which allows the visualization of antibody binding within cells or tissues. Flow cytometry, used to analyze the binding of antibodies to cell surface markers. RA, involving the use of radioactive tracers to measure the binding of antibodies to antigens in cell-based assays, where hybridoma cells can be screened for their ability to inhibit cell growth or function by targeting specific cell surface receptors or molecules. Step 6. Cloning and propagation of hybridoma cell. Hybridomas producing desired antibodies are selected and are then transferred into large culture vessels or flasks. The hybridoma cell lines are cultured using in vivo or in vitro methods. These hybridoma cells can be maintained and preserved in the culture media for the production of monoclonal antibodies. The figure here represents the steps in hybridoma technology. In vivo method for the cloning and propagation of hybridoma cell. In vivo method uses mice for the production of monoclonal antibodies. Mice are injected intraperitoneally with 105 to 110 viable hybridoma cells. After a few weeks, the ascites fluid is collected from an anesthetized mouse. Ascites fluids are contaminated with mouse immunoglobulins to some extent, and isolation of monoclonal antibodies requires purification. If purity of the antibody is important, then this technique may be inconvenient. In vitro method for the cloning and propagation of hybridoma cell. This is another method in which hybridoma cells are cultured in laboratory conditions. It involves growing the hybrid cells in a culture media followed by isolation of monoclonal antibodies from the media. This method is more suitable for the culturing of hybrid cells as it reduces the risk of contamination. In vitro antibody production leads to the production of highly pure antibodies. Applications of hybridoma technology. Diagnostics. Monoclonal antibodies are utilized in diagnostic assays, such as ELISA and Western blotting, to detect specific antigens with high sensitivity and precision. These assays play a crucial role in detecting diseases, identifying pathogens, and monitoring biomarkers. Therapeutics. Monoclonal antibodies are employed in targeted therapies for conditions like cancer, autoimmune diseases, and infectious diseases. Examples include trastuzumab for HER2-positive breast cancer and rituximab for B-cell lymphomas. Research. Monoclonal antibodies enable scientists to study specific proteins, cells, and molecular pathways with exceptional specificity. They facilitate research in areas such as immunology, cell biology, and genetics. Immunotherapy. Hybridoma technology has contributed to the development of immunotherapies, including immune checkpoint inhibitors and chimeric antigen receptor (CAR) T-cell therapies, which harness the power of the immune system to combat diseases like cancer. Vaccines. Monoclonal antibodies play a role in vaccine development, aiding in the identification and characterization of vaccine antigens. These are some of the FDA-approved monoclonal antibodies, which includes rituximab, Rituxan, trastuzumab, Herceptin, bevacizumab, Avastin, cetuximab, Erbitux, panitumumab, Vectibix, etc. Future aspects of hybridoma technology. Personalized medicine. Hybridoma technology can be used to create patient-specific monoclonal antibodies tailored to individual medical needs. This approach can revolutionize personalized medicine, especially in cancer treatment and autoimmune diseases. Next-generation antibody engineering. Researchers are exploring innovative antibody engineering techniques, such as the use of nanobodies and single-domain antibodies. These smaller and more stable antibody fragments offer advantages in terms of tissue penetration and modularity. Combination therapies. Hybridoma technology can be used to develop monoclonal antibodies that work synergistically with other therapeutic agents, such as small molecules, to enhance treatment efficiency. Targeting complex diseases. Researchers are exploring the use of monoclonal antibodies for complex diseases like neurodegenerative disorders. Novel approaches may involve the delivery of antibodies across the blood-brain barrier. Improved production and scalability. Advances in bioprocessing techniques are expected to improve the production of monoclonal antibodies, making them more cost-effective and scalable for broader use. Biosensors and diagnostics. Monoclonal antibodies generated using hybridoma technology will continue to be crucial in biosensor development and diagnostic applications, enabling rapid and specific detection of pathogens and biomarkers.